Good afternoon. Welcome. Welcome to our Meslem Center AD seminar series, which today is co-hosted by the Department of Radiology. And it's a real pleasure for me to introduce our seminar speaker today, Dr. Adam Brickman from Columbia University. Adam is professor of neuropsychology in the Taub Institute for research on Alzheimer's disease and the aging brain, and the Department of Neurology at Columbia. His research focuses on understanding the vascular contributions to cognitive aging and Alzheimer's disease in genetic, clinical, and community-based contexts. His work integrates neuroimaging and fluid biomarkers into observational studies, basic neuroscience, and epidemiological approaches. He is also interested in understanding sources of racial and ethnic disparity in Alzheimer's disease, developing interventions for cognitive aging, and designing neuropsychological instruments to assess cognition in older adults. Adam leads neuroimaging efforts in several large community and clinic-based studies, such as YCAP, the Washington Heights Inwood Columbia Aging Project, the YCAP Offspring Study, and the Alzheimer's Biomarker Consortium on Down syndrome. He's Associate Director of the Taub Institute at Columbia and Deputy Director of the Columbia University Alzheimer's Disease Research Center and leads its biomarker neuroimaging core. He has clinical expertise in cognitive aging, dementia, and neuropsychiatric disorders. Adam completed his undergraduate studies at Oberlin College, his PhD in neuropsychology at the City University of New York, his internship at Brown Medical School, and his postdoctoral training at Columbia University, where he has been on faculty since 2007. In addition, he has nearly 400 publications and is very well funded through the NIH. He's been awarded many honors, including the Arthur Benton Award for Mid-Career Research from the International Neuropsychological Society. So without further ado, I give you Adam, whose seminar is entitled, "Is There a Role of Cerebrovascular Disease in Our Conceptualization of Alzheimer's Disease?" Adam, take it away.
Thanks, everyone. Thanks, Bob, for that nice introduction. Can everyone hear me okay in the back? All good? Yeah, excellent. So thanks so much for coming to the seminar. Thanks so much for inviting me to Northwestern. It's a real pleasure to be here. I've been to Chicago many times. This is my first time at this medical school, and I've been to a lot of medical schools, and I think this is the shiniest and fanciest of them all, so it's really nice to see. So as you heard, I'm going to be talking a little bit about the role or the potential role of cerebrovascular disease in Alzheimer's disease. A lot of my colleagues in the Meslem Center don't need a primer on Alzheimer's disease, but I added some introductory slides for those of you who might not be familiar with some of the historical aspects of how we conceptualize Alzheimer's disease. So these are my disclosures, nothing specifically relevant except for funding, perhaps, to this talk. But I like to talk about my other disclosures, which is, like many scientists, I'm biased by and enamored by my own hypotheses. My salary is almost entirely supported by grant funding, which often motivates findings, something we don't talk about very much. And most of what I know I learned from social media. I used to put my Twitter handle here, but I'm off Twitter, I think, for obvious reasons. Now most of what I learned about neurodegenerative diseases come from articles that my dad clips out of the New York Times and mails me. So that's another great source of science. So I'll start with the takeaways, and I'll go through these 3 or 4 points throughout the talk. So first, I want to try to convince you that cerebrovascular disease contributes additively, which I think is obvious, but also synergistically and possibly directly to Alzheimer's disease pathogenesis, likely through tau pathology, and to its course in both late onset disease, or sometimes called sporadic Alzheimer's disease, and in genetic populations programmed to develop Alzheimer's disease. I want to talk a little bit about how integration of fluidic and neuroimaging biomarkers provide insight into the role of cerebrovascular disease in Alzheimer's and points to possibly inflammation and blood-brain barrier disruption, not just peripheral vascular risk factors, as potential drivers of these associations. And I want to talk about the role of cerebrovascular disease having implications for treatment of AD, both in terms of a target or an effect modifier, but also with respect to safety of some of these new trials. I'll end with some future directions. So everyone in the field of Alzheimer's knows this, but for those of you who aren't, Alzheimer's disease was initially, not coincidentally, described by Dr. Alzheimer, who had a patient, Augusta Dieter, and she had a clinical syndrome. She was a 51-year-old woman. She had reduced comprehension, memory impairment. She had aphasia, and she had some disorientation. She had some neuropsychiatric symptoms of paranoid delusions and hallucinations. And he diagnosed her with presenile dementia. Presenile because she was young, 51. I hope that's still young, considered young. And dementia because she had a progressive syndrome that got worse over time. So what was unique at that time was the emergence of modern histopathological techniques to appreciate the underlying cellular and protein abnormalities in the brain tissue. So Augusta D came to autopsy, and Dr. Alzheimer described the pathology that he saw as comprising intracellular fibrils, which we now know are neurofibrillary tangles. He described what are called miliary foci with peculiar substance, which we now call senile plaques. And the attending pathologist, although Dr. Alzheimer didn't make much of this, described pretty significant arteriosclerosis of the small cerebral vessels. And so he felt that he discovered a new disease marked by this clinical syndrome of dementia, together with these two protein markers on pathology. And it was his mentor, his boss, Emil Kraepelin, one of the fathers of modern psychiatric nosology, who coined it Alzheimer's disease in his influential textbook at the turn of the century. So it was considered pretty much a rare disease until I would say the late 70s, early 80s, it inflected a bit. And at the time, there were groups in New York, in Boston, in the UK, who started to understand that when we saw senile dementia, or what we used to call senility, in older adults, the pathology underlying that senility, which we thought of as a normal part of aging in many cases, was actually similar to the pathology we saw in this presenile form of dementia called Alzheimer's disease. And folks like Peter Davies, Bob Terry, Bob Katzman, and others concluded that this is a major health issue that affects not a few people with rare forms of early onset disease, but many, many people with late onset disease. And that formed, of course, many efforts like the Alzheimer's Disease Research Center programs that tried to get everyone on the same page with how we diagnosed and conceptualized Alzheimer's disease. And so a first set of diagnostic criteria that were put out in 1984, sometimes called the McCann criteria, was a probabilistic criteria set that was based on ruling out symptoms and potential causes of a dementia syndrome. So in the old days, we used to diagnose Alzheimer's disease clinically as probable Alzheimer's disease, and this was a dementia syndrome, and we would rule out any other causes or potential causes of the dementia. But of course, because we couldn't visualize the histopathology in vivo, it was a probabilistic diagnosis, and it was one of ruling out, not of ruling in. And then there were other categories of possible Alzheimer's disease, and definite Alzheimer's disease was only diagnosed when there was the presence of a dementia syndrome together with evidence of the histopathology of Alzheimer's disease, either from biopsy, very rarely, I hope, and autopsy, and obviously only after death. So I don't need to tell this crowd about the amyloid cascade hypothesis, but this has been the dominant pathogenic hypothesis of Alzheimer's disease in the field for a long time. It's very simple. In many ways, it starts with the accumulation of amyloid, which somehow gives rise or causes the accumulation of tau pathology temporally, and which induces or causes or gives rise to neurodegenerative or atrophic changes in the brain, and that's what is the proximal change to the cognitive symptoms and the clinical syndrome we see in the clinic. So one thing that's been exciting for me as I've grown up in this field over the last 20 years or so is taking these amyloid and tau and neurodegenerative features of the disease and now being able to visualize or approximate them in vivo using biomarkers. We can use PET imaging, radioligands to label amyloid and amyloid plaques and tau neurofibrillary tangles. These are two radioligands that we use in our lab a lot. We can use magnetic resonance imaging technology to appreciate the atrophic patterns in the brain and capture the neurodegenerative changes. And of course, now we can also measure abnormal levels of proteinopathies in the cerebrospinal fluid and more recently in the blood compartment. So if you've been to an Alzheimer's disease meeting, you've seen some version of this slide. These are the famous Cliff-Jack curves. And this describes the cascade of events that ostensibly happens in someone with Alzheimer's disease as they transition from being cognitively normal to this mild cognitive impairment phase to this dementia phase. And what you can appreciate is that in Cliff's model, it all starts with the accumulation of beta amyloid pathology, followed by tau-mediated neuronal injury, followed by atrophic changes. Sorry, I'm having a little trouble with my pointer here. Followed by memory decline, and then finally the clinical function goes down. And so there are a few things to point out here. What you can appreciate here is that in this conceptual model of Alzheimer's disease, the pathology accumulates years before the onset of clinical symptoms, right? And it still follows this sort of prescribed cascade of events. Now, these theoretical curves were translated into more modern diagnostic criteria for Alzheimer's disease, which basically look like this. So in 2011, those 1984 McCann criteria were revised into three categories. One category was preclinical Alzheimer's disease, where if someone had evidence of A beta pathology and maybe a little bit of tau pathology, they'd be considered to have preclinical forms of the disease. We had another diagnostic category called MCI, or mild cognitive impairment due to Alzheimer's disease. That was some cognitive impairment without functional impairment that was attributable and measurable with amyloid plaques and tangles. And then we had the Alzheimer's disease itself, which included biomarker evidence of amyloid and tau, together with the dementia syndrome. So we shifted from this rule-out diagnostic approach to this rule-in diagnostic approach, using biomarkers to capture some of the pathophysiological events and then define the disease that way. So these criteria were revised again in 2018. And in 2018, the task force that was put together by the National Institute on Aging and the Alzheimer's Association came up with these diagnostic criteria. If there is biomarker evidence they would first ask if there is biomarker evidence for amyloid pathology, and if there wasn't evidence for amyloid pathology, the person would not have Alzheimer's disease. If there was evidence for amyloid pathology, the next question would be, is there evidence of tau pathology? And if no, we would say that that person has Alzheimer's pathologic change, and if they did have evidence of both amyloid and tau pathology, we would say that that person has AD. So these are very different. Does anyone see the obvious thing that's missing in our evolution of the conceptualization of Alzheimer's disease? It went from a clinical syndrome to one that doesn't incorporate the clinical syndrome at all in the diagnostic conceptualization. Okay? So of course, at that time, the field decided that if you had Alzheimer's disease, you could still characterize our patients, our participants, as unimpaired, having mild cognitive impairment, or having dementia. So for the first time in 2018, we dissociated the clinical symptoms, the clinical syndrome of AD from the diagnostic conceptualization of AD. So these criteria were revised again in 2023. No flow diagram here. These are the new criteria. If you have A beta, you have Alzheimer's disease. This is a radical shift in our conceptualization of Alzheimer's disease. It's a little bit more nuanced. If you read the document carefully, there are these different pathophysiological stages that ostensibly reflect the cascade or the course of the disease, with amyloid positivity, tau negativity, et cetera. And then there are these clinical staging criteria that predict or characterize patients based on how much pathology they have in their brain, estimated through biomarkers, and what the offset of that pathology is with their clinical symptoms. Either they have less pathology at greater amounts of clinical syndrome, which would suggest some comorbidity, or the opposite, which would be greater pathology at lesser clinical symptoms, which might implicate a reserve or resilience type thing. I think this is great theoretically. I don't think we understand yet the relationship between biomarkers and the clinical syndrome enough to say who's on or off that offset. Anyway, so when I joined Columbia in 2004 as a fellow, a lot of my colleagues in epidemiology were doing these big community-based studies, and they kept showing, and others in the field, that all these vascular risk factors contributed to risk for Alzheimer's disease, or what we call probable Alzheimer's disease. Back then, including diabetes, insulin resistance, hypertension, atrial fibrillation. And that sort of leads to the question of does pure Alzheimer's disease, absent of vascular disease, exist? So let me go through this slide a little bit. These are from your neighbors at Rush. This is Julie Schneider's work of showing the pathological distribution of people who came from their community sample into their autopsy program. So they characterized their participants clinically as having no cognitive impairment, having mild cognitive impairment, or having probable Alzheimer's disease. And then they characterize the amount of the different pathologies that exist in those different categories. And that's what these pie charts represent. So V is vascular disease. 0 is no pathology. AD is pure plaques and tangles. AD plus V is plaques and tangles plus vascular disease, et cetera. And if you focus on this brown triangle, this is pure Alzheimer's disease, representing, I don't know, about 15% or so of people without cognitive impairment. And the Alzheimer's disease group represents about 35% of people with no cognitive impairment. If you look at the pure group, though, and you track across the diagnostic groups, it gets smaller and smaller. So people with pure Alzheimer's disease who have MCI, there's a smaller proportion with pure AD. And once you get to the symptomatic phase of the disease, maybe 5% or less of people diagnosed with clinical Alzheimer's disease actually have pure Alzheimer's disease. So one way that I think about it is that Alzheimer's disease, pure Alzheimer's disease exists, but primarily in people who don't have symptoms for Alzheimer's disease, right? So that's hard to wrap your head around, I think, conceptually. So I was interested initially in, well, you know, maybe this vascular stuff seems to be important. Maybe we can capture the sort of end organ damage in vivo that's caused by this vascular disease to see if we can characterize, the effect of vascular disease on the aging brain. It wasn't really a question about Alzheimer's disease at the time. We were interested in the white matter particularly, and the white matter is particularly vulnerable because the vascular supply throughout the brain is not uniform. White matter is perfused by mostly delicate vessels that are quite vulnerable to damage and pathology. There are other aspects of the vessels in the white matter. They're more prone to blood-brain barrier breakdown. We'll get into that a little bit later. There's some morphological features of the vasculature in the white matter. You can see that as the tortuosity of the vessel in the white matter, so as the vessels come in from the outside of the brain towards the white matter here, you can see that the morphology gets a lot more tortuous and they're more vulnerable to injury. So we're very interested in capturing the degree of small vessel disease in the white matter, and of course there are a lot of neuroimaging approaches to do that. We can look at the gross volumetry of the bulk morphology using T1 weighted MRI scanning. We can look at the microstructure using diffusion tensor imaging, or we could look at white matter hyperintensities seen here, and this started my love affair, 15-year love affair so far, or 20-year love affair with white matter hyperintensities, and I've been focused on these a lot. So to get to the story, cerebrovascular disease contributes both additively and directly to Alzheimer's disease. So like I said, we were focused on white matter hyperintensities. My lab developed ways of quantitating white matter hyperintensities very reliably. We have a patent on the software, and we've developed it over the years. And this is a single case of a single person and the degree of white matter hyperintensities they have. And these are the frequencies of white matter hyperintensities summarized over 30,000 participants in our and collaborative studies. So we can quantitate, it's quite prominent in the population. These are very, very common. A lot of the early work that I did and continue to do were in these 2 community-based studies, which you heard about in my introduction. These are older adults and middle-aged adults who live in the Washington Heights area of Manhattan. If those of you who've been following the news about New York, if you visit us, you don't have to pay for congestion pricing as you enter the city, because we're outside of the walls of that. So it's in the upper tip of Manhattan where tourists typically don't go. But it's a very, very diverse community. And most of the studies that we do focus on that diversity and try to capture the representation of the participants in our group. So this is an older slide. We've probably scanned about 6,000 people at this point from these communities. So we published a lot of papers early on. We first showed in many different ways that white matter hyperintensities as a marker of small vessel cerebrovascular disease predicts incident Alzheimer's disease. And this is just a survival curve showing that the people with a lot, or the upper third of amount of white matter hyperintensities, really fall off the cliff a lot earlier than people who have a lesser amount of white matter hyperintensities. We modeled latent change, longitudinal change in white matter hyperintensities as a function of change in diagnostic status. And we see that there's this increase, particularly in posterior areas of white matter hyperintensities, that goes along with the diagnostic phenoconversion from normal controls to MCI, or from MCI to dementia. We showed that among individuals who have evidence of amyloid pathology, if we characterize them as being symptomatic on the right side, or being asymptomatic, they all have the same amounts of amyloid pathology, but what distinguished the 2 groups was the degree of white matter hyperintensity or small vessel disease that they have. So it appeared early on that there's at least this interaction that if you had amyloid pathology, it wasn't quite as impactful on your clinical features unless you had white matter hyperintensities. And we showed that in individuals with amyloid pathology, particularly if they had a lot of white matter hyperintensities, they're at much greater likelihood of phenoconverting longitudinally from being a control to having MCI to having clinical AD. Next, we did some studies where we wanted to start to understand the impact of the vessel disease, or of the white matter disease on longitudinal outcomes. And so first we looked at tau measurements in the cerebrospinal fluid. And what we showed was that people with a lot of white matter hyperintensities, they had a greater accumulation of tau, increases in tau pathology, or tau protein levels in their CSF, compared to people with lesser amounts of white matter hyperintensities. That wasn't true in the reverse case scenario. In other words, tau pathology didn't predict the accumulation of white matter disease, but white matter disease predicted the increase in tau measurements. We also showed that white matter hyperintensities seem to give rise longitudinally to this pattern of atrophy in the cortex that seem to mirror in some ways some of the atrophic patterns that we see in Alzheimer's disease. And those patterns of atrophy mediated statistically the relationship that we see reliably between white matter hyperintensities and cognitive change. So we published all these papers. I'm sorry, I don't have a monitor, so I don't know what's coming next. It's a surprise to me, too. As you heard, I also have a very, very strong interest in disparities, and one of our hypothesized mechanisms or avenues that promotes disparities among different racial and ethnic groups in the incidence and prevalence of Alzheimer's disease is, of course, how much cerebrovascular disease that they have. We've shown main effects of race and ethnicity on lots of markers of cerebrovascular disease. These are white matter hyperintensities. And in this nice paper that Indira Turney did a couple of years ago now, she showed that when we combine our lifespan samples, we see an inflection in the accumulation of white matter disease with age in white people and in Latinx participants. But among our black participants, we already see this increase in rate of accumulation of white matter hyperintensities earlier on in the lifespan. So Indira, if you haven't seen this paper, you should check it out in JAMA Neurology. She wrote a very, very thoughtful discussion about the weathering hypothesis and how we embody the social stresses that go along with the socially constructed concept of race and ethnicity. So we wrote all these papers, and I was putting out paper after paper saying, white matter disease, Alzheimer's, hyperintensities are part of Alzheimer's disease. And I call this my hater slide. In more polite audiences, I call it my reviewer 2 slide. These are some of the comments that I've gotten over the years with respect to my grant applications and papers and stuff like that. Dr. Brickman, you're studying people with mixed dementia, not people with pure Alzheimer's disease, which is all that we really care about. Dr. Brickman, white matter hyperintensities or vascular disease in general is not related to amyloid, so it can't be part of the disease, right? So this gets into tautology, I would say. Dr. Brickman, white matter hyperintensities is totally related to amyloid. It only reflects the other form of amyloid pathology, amyloid angiopathy, the vessel distribution, or the vessel deposition of amyloid. Dr. Brickman, vascular disease is a part of normal aging, but AD is not. You're conflating normal with disease. I think I've already shown you that among people that we characterize clinically as normal, 30, 35, 40% of them meet diagnostic criteria for Alzheimer's disease. So what is normal and what's disease, I think, is still debated. Dr. Brickman, you don't know the literature. White matter hyperintensities are not a reflection of small vessel disease at all. They reflect something called Wallerian degeneration, or dieback of the axons from primary neurodegeneration. And Dr. Brickman, not everyone with AD has a lot of white matter disease. So these are the types of questions that motivate a lot of my research. They certainly wake me up in the morning, and they certainly have me shaking my fist at reviewers, but they're all reasonable critiques to try to address through our work. So after getting some of these criticisms, I started to think about, well, what's a good way of studying Alzheimer's disease, or the contribution of vascular disease in Alzheimer's disease, without the confound of, having high blood pressure, having a poor lifestyle? How do we isolate the fundamental, if there is a fundamental contribution? So at the time we were starting to think about some of these studies, there is a large study called the Diane study. I don't know if Northwestern is a site. It might be. This is a network of centers that see people with presenilin 1 or presenilin 2 or maybe APP mutations. These are genetically deterministic mutations for Alzheimer's disease, and they're fully penetrant. So if you have a parent with a presenilin 1 mutation, for example, and you inherit that gene, you have a 50% chance of inheriting that gene, and you have a 100% chance, if you do, of developing Alzheimer's disease. It's a perfect experiment. If we recruit everyone who has a first-degree relative or a parent with a known presenilin 1 or 2 mutation, they have a 50% chance by randomness, so they're essentially assigned randomly to either inheriting that gene or not inheriting that gene. And if they inherited it, they will develop Alzheimer's disease. So, and this was a landmark paper from Randy Bateman, and he did these initial analyses. These were called these pseudo-longitudinal data. So let me unpack it for you a little bit. A nice thing, or a convenient thing, I would say, about having a genetic mutation for AD is we can predict very, very accurately about what age you'll start developing symptoms based on the age of onset of your parent. So I'm 50 years old. If I inherited a presenilin 1 mutation from my mom, and she developed symptoms at age 54, we would say that I'm 4 years away from developing my symptoms of Alzheimer's disease. So we call that the estimated year to symptom onset. That's an important concept because it varies from person to person, but the scale is about the same. So someone who is 35 years old and inherited a gene, and their parent started seeing symptoms at age 39, they would also be 4 years to estimated symptom onset. So Randy's group used this concept to map all these different emerging biomarkers in Alzheimer's disease. And what you can see is they very much follow the Cliff-Jack curves. So in orange, these are folks who didn't inherit the gene even though they had a parent, and in blue are the folks who did inherit the gene mutation. And you can see for amyloid, at least, some 20 years prior to symptom onset, people start showing increases in amyloid pathology, at least here in the precuneus. We can see that if you look at total tau in the CSF, maybe about 15 years before symptom onset, people start accumulating tau pathology, and conveniently they used a really lousy measure of memory, but it made the point. to show that closer to symptom onset is where we start seeing the divergence in clinical symptoms. So we took data from the Diane study, and we asked the same question of white matter hyperintensities. Now, these folks are generally younger, and they're generally a lot healthier than our late-onset Alzheimer's disease patients, and they're essentially randomized into getting Alzheimer's disease or not. And this was work led by Sanju Lee. And Sanju showed that when we look at the total white matter hyperintensities, about 6 or 7 years prior to symptom onset, in gene carriers, we see this inflection, this increase in white matter hyperintensities. And when we look in posterior regions, it's even that much more dramatic. Up to 22 years prior to symptom onset, in young, healthy people without a lot of vascular risk factors, we start to see this increase in white matter hyperintensities, suggesting, at least to me, that there's this emergence of a cerebrovascular disease that's not attributable to hypertension, diabetes, et cetera, that seems to go along with the genetic mutation for Alzheimer's disease. We replicated these findings more recently with the Kalkiris's group. They have a similar cohort in Colombia, the country, Colombia, not the university, of mutation carriers. And we showed there, too, that prior to symptom onset, white matter hyperintensities increased and similar to other biomarkers of AD. And in that study, we also showed that the degree of white matter disease or vascular disease accounted for more variance in the cognitive outcomes than any of the other AD biomarkers. Okay, so I've been doing a lot of work in Down syndrome, and I'm very, very passionate about this work. For those of you who don't know, Down syndrome is another genetically predisposed population for Alzheimer's disease. It's trisomy 21 mutation. That's where the APP gene is. Triplication of APP causes a lot of amyloid deposition. Essentially, 100% of people with Down syndrome develop Alzheimer's disease. So I'm part of the ABCDS, or the Alzheimer's Biomarker Consortium. I run the imaging core and with Brad Christian, and we also have a separate R01 to look at cerebrovascular disease in Down syndrome. But I thought that this would be a very, very interesting population to look at with respect to vascular contributions to Alzheimer's disease. One thing that's very, very interesting about people with Down syndrome is although they do have morphological heart abnormalities, many of the other vascular risk factors are very, very, very low in people with Down syndrome. In fact, most people with Down syndrome don't have hypertension at all. Their vessels are also very, very clean. They don't have arterial atherosclerosis. They have a very low risk of clinical stroke based on their risk factor profile. So, of course, the question was, do we see increases in cerebrovascular disease as a function of diagnosis in people with Down syndrome? This was work done by my then postdoc, Patrick Lau. He's now a tenure-track assistant professor at Columbia. And Patrick looked at all of our favorite neuroimaging markers of cerebrovascular disease. He looked at white matter hyperintensities. He looked at enlarged perivascular spaces. He looked at frank infarcts that we painstakingly identified and quantitated. And then he looked at microbleeds, which, for those of you who don't know, typically when we see microbleeds, definitely in Down syndrome and in the aging population, if they're in posterior and cortical areas, they're typically considered a marker of cerebral amyloid angiopathy. So we looked at all these different markers, and not surprisingly, consistent with our hypothesis, surprisingly to my colleagues, we see this sort of dose effect by clinical syndrome stage of people with Down syndrome. So they all are sort of genetically programmed to get Alzheimer's disease, and the increase in white matter hyperintensities goes up as a function of their clinical diagnosis. And this is adjusting for age, adjusting for all the covariates you can imagine. We saw similar patterns, that the numbers were a little thinner in this paper, but they also surprisingly had frank infarcts. And I need to get to the bottom of this, because I was told that people with Down syndrome never get strokes or infarcts, and we definitely saw infarction, and I have a suspicion they're actually very, very large perivascular spaces, but that's a different discussion, which we also see in large perivascular spaces, and we certainly see a lot of microbleeds. Patrick, in a subsequent paper, this was just published last year, tried to model the inflection point. If you think about Down syndrome as a time to event syndrome, a time to event to developing Alzheimer's disease, we can model as a function of age where we see that inflection in different biomarkers. And what he found was when we look at these different cerebrovascular markers as a function of age in people with Down syndrome, they inflect at about age 35, 31, 36, 31, depending on which marker you're looking at, but generally in the early 30s. And if we compare this inflection point to PET markers of amyloid and PET markers of tau outside of Braak stage 1 and 2, what we see is that the vascular lesions that we're measuring come on board, inflect contemporaneously or even prior to some of the Alzheimer's pathology that we're measuring with neuroimaging. So this, in my mind, helps, doesn't prove, but it helps address the issue of the temporality or the causality between Alzheimer's pathology and the emergence of vascular lesions. If the vascular lesions are preceding or contemporaneous with the Alzheimer's pathology that we're measuring, they necessarily can't be the result of the Alzheimer's pathology, which is one of the hater comments that I frequently get. In a animal study that was led by a former student, Crystal Lang, in my lab, and with collaborators at Downstate University, we created a mouse model to try to approximate the white matter lesions that we're seeing in human imaging. And what we did was we occluded the MCA. We did transient occlusions for 30 or 60 minutes and then reperfused the animal. So these were just brief occlusions. We hypoperfused one hemisphere and then reperfused it. And they don't cause massive strokes, as you might imagine. They cause really subtle changes in the brain's white matter that we proved to ourselves that we could stain the white matter and see the types of disruption we would expect in underlying a white matter hyperintensity. But what was interesting was that we also detected phosphorylated and total tau levels that were increased in the ipsilateral hippocampus. So inducing hypoperfusion in these wild-type mice induced tau pathology in those mice without any genetic mutation for Alzheimer's disease. So we could come up with a model that we can induce the later pathological features of Alzheimer's disease simply by hypoperfusing the mice. This resulted in a grant application that was unfortunately not funded, and then which I submitted with my good friend and colleague Herman Moreno, who passed away very suddenly from pancreas cancer. So it's work that we're looking for the right collaborators to take up again. So the second point of the story is that if we integrate fluid and neuroimaging biomarkers, I think this can help us provide insight into the role of cerebrovascular disease in AD, pointing to blood-brain barrier and inflammatory markers. So this was, again, work in the Down syndrome folks. This was a student of mine, Famita Moni, who put together this paper. And I'm not a huge fan of omics-type analyses, but my colleagues convinced me to do a sort of proteomic analysis. And we essentially used some machine learning techniques to associate some of the markers that we see in the proteome with some of the cerebrovascular lesions that we're measuring on MRI scan. And in asymptomatic folks with Down syndrome, we see correlations of these vascular lesions that seem to point to this inflammatory cascade, a lot of CRP, a lot of interleukins, the adhesion molecules, VCAM and ICAM. And as the participants go into late-stage disease on the right, we start to see more of a relationship between those cerebrovascular lesions and the A-beta and tau and neurofilament light chain markers in the blood. So we've been doing a little bit of what's called TSPO PET imaging. This is ostensibly a reflection of microglia activation, and we're starting to see, this is very, very early, that there is this co-localization between posterior amounts of white matter hyperintensities and this increase in microglia activation. And this is across different patient subtypes and normal controls as well. I collaborate very, very closely with Donna Wilcock at Indiana and Sabrina Samoas, who's at Columbia. And Donna, as part of the MARC VCID consortium, has been putting out, together with her colleagues, these different markers of these biomarkers for vascular cognitive impairment in the plasma. And she's been really focusing on this VEGF family of proteins that seem to be somehow regulating angiogenesis and involved with maintenance of the blood-brain barrier. And in her studies, this placental growth factor, which is a very, very interesting marker, seems to be one that captures, preliminarily at least, some degree of blood-brain barrier dysfunction. And so Natalie Edwards, who's a doctoral student in my laboratory, has been trying to integrate these fluidic biomarkers into our pathways of linking vascular changes in the brain with Alzheimer's disease type changes. And using these are cross-sectional data, but she used path analysis statistically, and what she showed was that it appears, statistically at least, that white matter disease gives rise to GFAP, which is a marker of astrocytosis, which gives rise downstream to p-tau, marker of tau pathology or tau changes, which gives rise downstream to neurofilament light chain, which is a marker of neurodegeneration. So the model that fit the best in these cross-sectional data was one in which vascular disease affected the astrocytes, which had downstream effect on tau and neurodegenerative pathology. And she's been modeling these in all sorts of different ways. We linked in the PLGF. This is not in Down syndrome in a community-based study. And what we found was that PLGF, as a marker of blood-brain barrier dysfunction, again, statistically, was upstream from GFAP, upstream from white matter hyperintensities, and that gave rise to p-tau and NFL changes or variants. And so this path analysis, again, cross-sectional, again, statistical, not causal, but it's consistent with the hypothesis, or most consistent with the hypothesis, that blood-brain barrier dysfunction gives rise to white matter disease, or a certain degree of white matter disease, and to astrocytosis, which has downstream effect on Alzheimer's disease risk. So these are data that we're putting together now, and we're hopefully writing it up very soon. I'll nag her about that when I get back. So finally, the role of cerebrovascular disease, I think, has very important implications for treatment of AD, both in terms of target, as a target for treatment, but also for effect modification and treatment. And we took data from the A4 trial, and we showed that in these very well-screened individuals that the amount of white matter hyperintensities correlated almost as strongly with the main outcome of the trial as did amyloid pathology itself. So we're seeing that even if we want to ignore vascular disease as causal in Alzheimer's disease, it's accounting for almost as much variance as the clinical outcomes in Alzheimer's disease as amyloid pathology is itself. And then I've been working with colleagues at Harvard to understand whether baseline white matter disease and other markers of cerebrovascular disease predict who in clinical trials is going to develop some of these adverse outcomes, namely edema and hemorrhagic type lesions. Okay, so some final thoughts. So I think I tried to make a case that, there appears to be some sort of an endogenous vascular component to Alzheimer's disease risk pathogenesis and course that can't be just attributed to vascular risk factors. White matter hyperintensities in the context of Alzheimer's disease are not just a reflection of supraamyloid angiopathy. We can do that by controlling for the amount of microbleeds, for example, or of Wallerian degeneration, because the spatial and the temporal emergence of white matter disease doesn't follow the spatial and temporal emergence of neurodegenerative changes. In fact, it's the opposite. I think a huge focus of this work should be on the neurovascular unit on blood-brain barrier dysfunction, which we're certainly pursuing. And as I mentioned before, vascular risk factors may exacerbate these AD-related cerebrovascular dysfunction that we're seeing. So it's not good to have hypertension and diabetes. That could certainly amplify what might already be going on that's specifically related to Alzheimer's disease. And so I put Sandra Black, for those of you who know her, she's big on Venus, on the Venus system. So this is my nod to Sandra Black, that we talk a lot about the arterial system, but very little about the venous system. So some approaches that we're taking, we've been trying to implement this blood-brain barrier imaging approach in the lab. This is what's called a motion-compensated diffusion-weighted gradient and spin echo pseudo-continuous arterial spin labeling sequence, which essentially uses water and diffusion and ASL sequences to estimate blood-brain barrier dysfunction. This is work that Danny Wang has pioneered, and he shared his sequence with us. And so far, we're getting a little bit of signal. If I didn't have this blob superimposed on a brain, it would be a brainish shaped blob. We're not sure exactly what we're measuring yet, but we're starting to see some interesting relationships between this KW exchange rate as a measure of blood-brain barrier dysfunction and some things that we would expect to be related to blood-brain barrier dysfunction. I work very, very closely with Liz Head at UC Irvine and in our Down syndrome grant that we have together. As folks pass and we get their brains, we're doing a lot of histological studies on their brains, and what we're starting to see is certainly co-localization of fibrin as a marker of blood-brain barrier dysfunction and amyloid pathology, but there's so much more that we're starting to see in these data as they come in. We've autopsied about 30 participants to date, and so this is a major, major focus of the lab right now. And I'm going to skip this just in the interest of time. And finally, I think that we've been discussing here about, amplifying and the neuroimaging core at Northwestern, and I wanted to just highlight some of the tools that we've been developing in our core that I think are going to be very, very useful to get a comprehensive view of the aging brain in terms of its vascular function and pathology. And so this 12-year-old looking MD PhD student, Anthony Cheesebro, in my lab, he's moved on, but he came up with something that we call MAGIC. Don't ask me what it stands for, I forget. But this is a semi-automatic way of detecting cerebral microbleeds in the brain by using gradient echo MRI or susceptibility weighted MRI. And this is going to be very important in ARIA-H or amyloid-related imaging abnormalities of the hemorrhagic variety in certain clinical trials. We work closely with Jose Gutierrez, who's a stroke neurologist, in quantitating perivascular spaces. Right now we do it visually, but we're using some machine learning to try to identify and quantitate these markers automatically. We've implemented phase contrast imaging to look at extracranial blood flow, into the brain at the level of the internal carotid and vertebral arteries. And of course, we're doing a lot of ASL imaging and diffusion weighted imaging in the lab as well. White matter hyperintensities are great. They've been around for a while, but there are lots of ways of quantitating them. So Mohammed Al-Shiko, a senior engineer in my lab, has been using texture-based analyses to try to get at the heterogeneity of the signal in the white matter hyperintensities to understand the underlying pathology and its correlates. And so he's been working very, very hard at coming up with some new paradigms to capture different aspects of the heterogeneity of signal, of the sphericity of the lesions, of the proximity to the ventricles, et cetera, to get a much more comprehensive, multidimensional view of white matter disease. And he's also built this incredible neuroimaging core workflow, which we've been working on for the last couple of years, which is essentially a one-stop shop in my lab where it goes from uploading a DICOM image to analyzing, QCing, and databasing the outcomes all through GUI interfaces. And we've just finished building this whole system that's been working very, very well. And the last thing that I'm super excited about. I was hoping that we'd have the notice of award by this visit, but we haven't gotten the NOA yet. But we've been very, very interested in ultra-low field MRI scanning. This is, in fact, an MRI scanner. It's made by Hyperfine. It is 0.07 Tesla in field strength, which is about 70 kitchen magnets, if I Googled that right. And we can get incredible images with the magic of AI and lots of recording. And so if we get this grant, which I suspect we will, we're going to be deploying this Hyperfine system in rural communities in South Africa and scanning about 5,000 people over the next 6 years that are enrolled in a large epidemiological study of cognitive aging and dementia in South Africa. So this hopefully will get started within the next couple months, and hopefully I'll have some cool stuff to show you at another time with respect to this project. This is just an example of some neuropsychological testing that's happening in the field, and that's my colleague Dejania, who's set up the high field MRI study that we already have ongoing there. So with that, I just want to acknowledge my current lab members, some past lab members, many of whom have gone on to do much cooler things than when they were working with me, but they really do all the heavy lifting here. I want to thank many of my collaborators, some familiar names here. My closest collaborator is Jennifer Manley. Richard Mayu has been my chair collaborator and superego in many ways, and some very, very close other friends and colleagues on the list there. So this is a recent picture of the lab, and thank you so much for your time and attention. I'm happy to try to address some questions if we have time.
Wonderful. Great talk, Adam. So we have time for a few questions here. We're going to have some microphones because people are on Zoom, we want to make sure that they're able to hear the questions. So, Marcel.
So, Adam, I don't know how easy it is for you to go back to the slide where you had a bar graph of white matter intensities in Down syndrome by normal, MCI, AD.
Let's see. You can't seem to exit out of this one, or?
No, yeah, that one. So if you look at the definite AD, there's a tremendous amount of spread.
Yeah.
So have you had a chance to look at the top three and the bottom three and to see what differentiates them?
Yeah, it's a fantastic question. The spread, the numbers are much larger now, and the spread remains. We see a lot of variance, and so we haven't yet, but that's certainly a big question. There might be some genetic modifiers, which would be a hypothesis, or all sorts of interesting genes on chromosome 21, but it doesn't appear to be related to most of the lifestyle factors that we would guess. It's not hypertension, not diabetes, certainly not high cholesterol. Sleep apnea seems to play a little bit of a role, but it doesn't account for all of this variance that we see. But it's a great question, great observation. Yeah. Oh, sorry.
Beautiful talk. Thank you so much. You know, as a behavioral neurologist, I see a lot of cases with these white matter changes. This is very common. And then many times we can't find the actual cause of the white matter changes. So per your experience, if you have to give a differential of what the white matter changes are with aging, which etiologies do you put on your, top of your differential?
I think it's probably heterogeneous. I mean, obviously, if someone has a history of hypertension, diabetes, overweight, high cholesterol, certainly that's accounting for a lot of what we see. In the absence of peripheral disease, I suspect there's something inflammatory happening that's accounting for disruption at the level of the neurovascular unit at the endothelial level, and I think that's yet to be determined. For years, I was doing longitudinal type analyses, and we'd have a frustrating, small but notable group of people whose white matter hyperintensities seem to improve. I used to have a slide that I called white matter hyperintensity axioms, and the second point was they only get worse, they never get better. And I wasn't sure if this was just measurement error or real. And I think I'm convinced that we actually do see some regression of white matter disease, which would explain something more transient, something inflammatory, or,
Sorry, I think I should have framed my questions a little different. If you rule out all the, major causes of white matter changes, like, vascular hypertension, in those cases, do you think Alzheimer is high on the differential? Is it, do we need to think about Alzheimer seriously in these cases?
I wouldn't use white matter hyperintensities as diagnostic for Alzheimer's disease. And that's an important point. It's very, very, very common. And certainly, maybe with our texture-based analyses, we might find a signature that's specific to Alzheimer's disease. I think that the point that I'm trying to make is using white matter hyperintensities as a tool to probe sort of end organ vascular damage, to ask the hypothesis of whether there's a vascular contribution to Alzheimer's disease, but it's not diagnostic for Alzheimer's. That's an important case, particularly to people who've had an MRI scan and know that they have a lot of white matter hyperintensities, which is very, very common, of course. Yeah. Farzaneh?
Yeah, Adam, so sorry, I was jumping in because I wanted you to stay on the slide.
I'm sorry.
Side, but you don't have to go
Okay.
back since we've seen it again. But I think, so you had on that x-axis definitive Alzheimer's dementia.
Right.
You know where I'm going. So is that the sliver of people on the post that only have AD pathology?
No, that's a great question.
Then why do we call it AD dementia?
Because this is in Down syndrome.
And
right?
not mixed dementia?
All dementia is mixed dementia. Sorry.
No, but I mean, there is that sliver, right? A small 20%.
Yeah, mostly in asymptomatic people. So I would say that they.
But you had it in that slide.
It's
right?
like 5%.
5%. So 5%. So sort of, because I think throughout this, we talk about Alzheimer's disease and then clinical Alzheimer's disease. And then I just want to know what we're studying.
So first to specifically address the question, the definitive AD dementia group, that was in the Down syndrome folks. And when they meet criteria for dementia, unless they've had a major head injury or something metabolic, we call that Alzheimer's disease because they have genetically deterministic Alzheimer's disease. In other words, we're not wondering if this is AD or Lewy body dementia or FTD. They have AD, right, when they're demented. I'm trying to make the point that in this pure genetic form of AD, they also have mixed dementia. So even in these pure cases, presenilin 1, presenilin 2, Down syndrome, they have mixed pathology. So if we were at a bar and drinking and you asked me what I think Alzheimer's disease, should it be defined by plaques and tangles, I would say no, it's a syndrome that comprises mixed dementia, mixed pathology. I'm not sure if you seem skeptical, but we can talk more, yeah.
Yeah, Adam, the folks at BU have done extensive studies on aging primates and have seen white matter changes that correlate with cognitive deficits. Yeah. And so do they get white matter hyperintensities?
That is a great question. It's Doug Rosine's group, right, and others, right? So you and I may have been on the same study section when we reviewed one of those grants. They proposed it in a grant application, I can say that with confidence. I don't think they ever reported it. In their monkeys.
Because those animals clearly don't get Alzheimer's disease.
Right.
I'm just wondering, is the vascular disease medicine effective against AD or Taub pathology?
Do treatments for for
medicine against the vascular disease.
Yeah, so if we control hypertension aggressively, will we see a reduced risk for Alzheimer's disease? Is that what you're asking?
Yeah, I'm just wondering, it's a clinical case or data.
Yeah, so the SPRINT MIND trial, I thought showed some pretty compelling data that if we aggressively control blood pressure with blood pressure lowering medications, we see a lower incidence of dementia and MCI. They couldn't call it Alzheimer's disease because they were clinically diagnosed. Do you have, I sensed Farzaneh was going to chime in because you. So I think that there have been a few indicators that aggressive blood pressure control, for example, will help dementia outcomes, whether it's specific to Alzheimer's. I don't think anyone's tried to treat hypertension in people with AD dementia per se. And I'm not sure that would be the right treatment either. I think it'd be more inflammation type stuff that would be more relevant.
Okay, I think we should give Adam another round of applause for a great talk. Thank you, Adam.